please.
Can everybody see this?
Nope, we're gonna go ahead and.
I think that was pretty good.
Yeah, that is good.
Okay, perfect. Well, hello everybody. Thank you so much for coming and thank you so much for having me. My name's Rebekah Pettit and today I'm going to be talking to you about the Mangrove Saltmarsh Snake, Nerodia clarki compressicata. So before I get into the details of the presentation, I just want to tell you a little bit more about me and why I'm here today and what I'm doing. So I'm a recently graduated biologist from Palm Beach Atlantic University, which is a small private university in West Palm Beach. During my last year completing this degree, I was able to participate in a reptile conservation research study with Dr. Thomas Chesness and Joshua Holbrook, who happens to be one of the founders of this group, so that's pretty cool. I got my degree in a Bachelor's of Science of Biology that was concentrated in zoology. So during this last year, it was really a privilege to study something that was in my field. Upon graduating, I'm currently working for Reptiles Magazine, which maybe some of you have heard of. Reptiles Magazine is all around reptile and amphibian enthusiast magazine. We have articles, care sheets, news topics, and just everything about reptiles and amphibians, from the educational point to the hobbyist perspective. There's vet articles and so much more, so I definitely encourage you to check that out. You can see at reptilesmagazine.com where you can subscribe to the magazine. Along with this, I also am a content strategist for the company where I run all of their social media. So you can follow us on Facebook, Instagram, and Twitter at Reptiles Magazine, these links right here. So I definitely encourage you to do that because it's, our care sheets are used by some AZA zoos and other facilities that are all about husbandry, reptiles, and amphibians. So kind of extending this topic for social media, I also run my own personal reptile blog on Instagram as Venomous. And this is more of just like a personal page to showcase animals that are in my life, specifically reptiles. So if you'd like to follow more of a personal journey and learn about me there, you can always do that and see some of the animals that are in my life. But this is really just the tip of the iceberg for what I would like to do in, as a career. I'd really like to contribute to reptile conservation. So it's a super privilege to be able to talk about some of the studies that I was a part of, but it's really just the beginning for me and what I'd really like to do. But today I'm going to talk to you about saltmarsh snakes, specifically the mangrove saltmarsh snake. Today I'm going to talk to you about the conservation status of the species. I'm going to talk to you about some of the field methods that we used that worked, and I'm going to talk to you about the threats, and also a very brief summary of some further research I did in a semester project that was required for my degree. So Nerodia clarki compressicata, or the mangrove saltmarsh snake, is an endemic species to the coastal aquatic habitats of Florida. These saltmarsh snakes, there are 3 subspecies. So we have Nerodia clarki compressicata, the mangrove saltmarsh snake, Nerodia clarki, which is the Gulf saltmarsh snake, and Nerodia clarki cineata, which is the Atlantic saltmarsh snake, and that one is a protected species. For the research that I was involved with, we focused on the mangrove saltmarsh snake, which you can see is the red snake there, but we also did a couple surveys where we looked at the Gulf saltmarsh snake, and that is this striped one over here, which is more in the northern areas of Florida. So this is a very understudied species in general, all of them. Not much is known about what they're doing or their lifestyle habits. They are an estuarine species, so they like to inhabit brackish waters, which is just a mix of fresh and salt water. So that's the mangroves, the marshes, and the wetlands that are all a part of the natural habitats of Florida. So focusing on the mangrove saltmarsh snake is really, was really the focus of our research. Again, they are a estuarine species, so they inhabit the brackish habitats. They are a species that is semi-aquatic and semi-arboreal, which just means that they enjoy the treetops as much as they enjoy the water. They are, again, endemic to the coastal habitats, so that is really where we were surveying and what the research really aimed to do. So when it comes to the habitat that they prefer, again, there's not much known about the species, but they really seem to prefer this high density vegetation areas. This includes, again, the marshes, the high grasses, and really the density of the mangroves and the prop roots, as you can see here. This is a very typical area that we would find them, and really is the ideal habitat we were looking for because it's a very hard area to survey for. It's very lush and dense, and it's hard to get through. So this is really the ideal area that we were looking for during our research, and this is definitely the preferred habitat of this brackish species, regardless of which subspecies. So when it comes to the feeding and habitat of the species, they are documented piscivores, and that just means that the only thing they've ever been documented eating is fish. But since they do inhabit such a diverse area, we did hypothesize that they could be eating other things opportunistically, such as birds, gastropods like snails, maybe crabs, or other invertebrates. But as of now, they've only been documented as a piscivore. Here are just some photos showing you them in their native wild habitat that I took some pictures of. Again, they inhabit the treetops comfortably, the marsh grasses over there, as you can see the Gulf saltmarsh snake doing, and down here in the prop roots is really where we typically found them in almost all of our surveys. So this species is super understudied, almost nothing is known about them, and that makes their population distribution really patchy. So it's almost impossible to know where these populations are and what's happening to them. So among that, they also inhabit this really diverse, really thick area that makes them, really hard to find, and so that makes them experts at being elusive. So when it comes to their hunting, we had to really strategically time when we were going to survey, and this was also very influenced by the tide. Since they like to go in these very dense areas, the fish, they become trapped, and that's when we really found the most. But it's very dependent on the tide, so we took that into consideration during all of our surveys. So the way that we went about this research is to survey state parks in Florida that they've never been documented before. Since their population is really patchy, there's a lot of areas where we just don't know if they could be, but there's the right habitat. So that was what really directed the way that we organized our study. Since these snakes, again, they're not established, we don't understand their numbers or what's happening to them. So this research was a presence-absence study where we simply just went to survey the areas, state parks specifically where there was habitat and they had never been documented, and we would do a presence-absence survey. So I'm just going to talk to you a little bit about the methods that we used and what was successful. So a little bit more detail about the snake that makes it really cool is that they come in these natural color variances. There's red, black, silvers, and greens. This is all natural, not human manipulated whatsoever, and these snakes also do face hybridization threats, and I'm going to talk a little bit about that as well. But here's a red-faced Clarkeye, and then on the next slide I have a really cool poster that was created by Joshua Holbrook that just shows some of the natural color variation that the species comes in. So this is all the same exact species of specimens we found during this study, and they were taken and created this poster by Josh. So this is just showing that. So what we hypothesized is that if there was habitat, there would be Clarkeye. So this really dictated the way that we went about our study. We would look at the areas through a Google map first, and then go there during the day to survey to see kind of what we were walking into before it was nighttime. This species is nocturnal, so that was we had no option really in when they're active. So we would have to go back during the night. Hopefully the tide was right, and that's when we would find them, or we wouldn't. This is really the ideal habitat that we were looking for and what we'd be hiking through for the whole night, basically, around this edge. This is the areas where they like to hide, and they're ambush foragers. So that just means that they sit and they wait for their food to come to them unless they really have to go and look for it, which we don't, again, really know what they're doing. So this is really the areas that we were looking for. We would survey in the day and come back at the night to find them. Again, these were done in state parks in Florida, all in areas where they had not been documented before. Some of them they had been documented, but only a couple specimens, so we went back a couple times during the year. But these were all done under FWC and state park permits for nocturnal collecting and also scientific collecting. So if you plan to be looking for nocturnal reptiles and snakes like this, I always encourage just to check your local laws and the rules of the parks. So although we quickly found that simple foot hiking and capturing them by hand was the best method for the species, we did go about some other methods to try and capture them. So I'm going to just briefly go over those as well. The first, during this program that I was able to be a part of for the research, we were able to intern with the Environmental Protection Agency at a couple different state parks. We surveyed MacArthur State Park, Sea Branch, as well as Grassy Waters Preserve, doing different herpetological methods to hopefully capture reptiles and amphibians in the park. So one of the first methods we did in Grassy Waters State Preserve was aquatic trapping. These are just simple basket types of traps that are connected with hooks and thrown in with ropes, and we would put 10 to 12 out and leave them for the day or night. They would be in shallow enough areas so there was air, so if anything got in, they wouldn't drown if they needed to leave. We would go back and check the next day. So this is just a photo of me being really excited about this giant siren salamander that was in one of the traps. I never saw one of those before, and it was a couple feet, in length, so it was really cool. We didn't find our target species with this method, but we actually did get a freshwater snake species, so that was pretty exciting. But we did get a lot of siren salamanders and newts and some frogs as well. So it definitely is an efficient method to survey for amphibians and some reptiles. The next method that we used was dip netting, and we did this in John Dickinson Park as well as Grassy Waters Preserve. The dip netting is just a really simple method of taking like a giant net that has small openings and going to the bottom of a shallow pond and just moving it along to see what you could catch through that. So we were hoping to maybe get some small snakes, but no snakes were found. We really focused on either ephemeral ponds or fish-dominated ponds. The ephemeral ponds is just a fancy word for ponds that have no fish in them. This is a really special natural habitat of Florida, since we are the wetlands and we are the flattest state in the country. Our habitats really rely on what's called microtopography, so that's just very slight elevation changes in the ground. And because of these areas that are created by trees or some of the marshes and the wetlands, these places that are usually dry during the wet season, they will fill up to be actual ponds. And these ponds are special because they don't have fish in them. So this is a really important habitat for amphibians because the fish aren't eating the tadpoles. So when we were dip netting, we would do this, we did this for 2 weeks. We did about an hour in each pond, whether it was the ephemeral pond or the fish-dominated pond, and hoping for our target species. During the ephemeral ponds, we got multiple species of tadpoles like pig frogs, leopard frogs, and also spadefoot toads. When it came to the fish-dominated ponds, we didn't even find one tadpole. So it shows you the drastic differences between these areas and how fragile they are. It's also a transitional habitat, so these animals who start off in the water and then they go to land or they come back. It's an important area for the amphibians, but also other animals like mammals, like deer and raccoons, they drink from these freshwater depressions. So it is a very important area, and something I found really interesting that's also facing threats, because in the past people have stocked these areas with invasive fish. For whatever reason, personal or commercial, it definitely disrupts the ecosystem, and of course it's very illegal. So it's a special habitat of Florida, so I thought it was of notable mention. The next trapping method that we used for reptiles was pitfall traps, and it simply is basically a fence that's set up throughout the park in a specific area for a specific length. And within this fence, there are buckets that are dug into the ground. So whatever crosses throughout the night, it will fall into the bucket, and then the next day we would go and check it. Within these buckets, we had a PVC pipe and also a sponge that was always wet. So in case it rained and it got flooded, they would have something to hold on to and float on. And if it got too hot, they would have something to stay hydrated. So we were checking these every day. This was in Sea Branch as well as MacArthur State Park. We didn't find our target species, but we did get some species of snake and also this gopher tortoise one day, which was really exciting and kind of funny, but he was okay. So we were definitely checking these all the time, as it's really important, because if you just left it, they could die pretty bad. So although we didn't find our target species through any of these methods, here are some of the faces of the other native species we came across through these methods. So it's definitely an effective way to survey for reptiles and amphibians. And usually these state parks, they're not really doing this actively. This was a part of the summer internship program that I was a part of. And we got to contribute to the state park species list because a couple of these species had never been documented in the park before. So although it wasn't our Clark Eye target species, we did find some cool reptiles and amphibians. So we have like a land crab here. Those are the spadefoot toads. There's a skink and also the scarlet snake. So these are all native species that I found really cool that we came across. So again, the most efficient method for our target species was simple foot hiking and capture. We would come back and survey sometimes all night and just grab these snakes by hand. They would be just sitting among the vegetation, and we just grab them and bag them, and then usually do our data and morphometrics right there in the field for that night. So you can see a picture here of me doing that with, I had 2 other student researchers that were in the program with me as well. So I'm just going to talk to you a little bit about the morphometrics that we did and how we analyzed our data a little bit. So the morphometrics included sexing the animal, weighing the animal. We did something called palpative induction, which I'll explain a little bit more later, as well as scale counts, measurements, as well as data collecting in our notebooks and just keeping documenting everything. So when it came to sexing them, we just checked if it was male or female by probing them. We would weigh them with a scale. We had like a whole kit that we would bring out with us. Palpative induction just means you basically stimulate the snake's stomach to see if it ate anything that didn't digest yet. You can almost squeeze them like a toothpaste tube, and if they haven't digested anything, it doesn't hurt them and it will come right out. Again, because this species is only documented as a piscivore, finding anything else that they would be eating is kind of a big deal for the species understanding. So we would always do that, and we only found fish. So there was no other animals that we found that they were eating, but we definitely think it could be a possibility. The scale counts was a really important part of the morphometrics because this is how we determined the purity of the species. It's a very old school technique, but it was efficient for what our data was because we didn't do any genetic analysis. So basically you go to the midline of the body of the snake and you do scale counts, those tiny little scales, where 19 to 21 indicated it was a hybrid, and 22 to 23 scale counts indicated a pure compressicata species. So again, that is the subspecies we were focusing on for this research, but there were a few areas we surveyed that had the Gulf saltmarsh snake. But overall, both really understudied, so anything that we found is helpful to their understanding. When it came to the data collection, we just documented the location, the weather conditions, colors, and types of things like that to keep our data organized. So when it comes to hybridization, this is a big threat to the species. I put this first because generally in conservation studies, hybridization is a bad thing. So why is it bad? Basically because it compromises the genetic purity of the species. So by hybridizing over generations, eventually the purity of the compressicata could be jeopardized and lost to extinction. These guys hybridize with freshwater snake species, Nerodia fasciata, and this is more, they don't like the saltwater. You never really find them in the saltwater areas, so because of the habitat loss that the mangroves and the estuary systems are facing, we hypothesize that it's pushing these populations to areas that they're not usually going to be found or usually live. So by overlapping with these water snakes, that's what's causing the hybridization. So we were able to determine that by our scale counts, and sometimes there were some exceptions, but overall this was an efficient way for us to determine what was hybridized. Also the hybrids, they're a lot more foul-tempered, they like to bite. The freshwater snake fasciatas are kind of mean, but the compressicatas are actually pretty docile for a water snake. So when you pick them up, most of the time they don't even try to strike you. So that was a big indication as well. So these patchy numbers, helping to understand them was a really important aspect of our study, and really is the whole focus of the presence-absence research. Because without understanding where these populations are established, it's really impossible to understand the impacts of their loss, especially as a predator on a system that's so diverse like the mangroves. So some of the successes of this research, although it is an unpublished research study, it is ongoing. We've been surveying for a year, but our permits ended in July. But we did document the first species in Broward County as well as Martin County. Dermot was actually there with us on the day we found it in Broward County. So both of those findings did get published in those local newspapers, which was pretty cool. They actually have not been documented in Palm Beach County as of yet, so we're still searching for them, although the permits have expired and the grants that were funding this research also expired, and I'm already graduated. I definitely made a lifelong connection to this research team and enjoyed going out there and herping with them and looking for this awesome snake. So we're still searching for it in Palm Beach County for sure. But again, understanding these population numbers was the focus of the conservation research, because without understanding where they are, it's impossible to plan for development, it's impossible to plan for conservation, and again, it's impossible to understand the impacts of their loss, especially as a predator. So that's really the focus of the research and what I was able to participate in as an undergrad. It was kind of something special that this university offered, and I took the opportunity to apply to it because it really helps expand your resume, give hands-on field experience, and also being part of a scientific team. It was also cool to be the only female on the team of guys to represent women in herpetology too, which I think is important. And it's definitely something I would recommend if you'd like to pursue a field that's competitive and underfunded. That you really need to stand out and get as much experience as you can. So the fact I was able to do that with my education is something that was special and definitely a privilege from Palm Beach Atlantic that was awesome, along with Josh and Dr. Chesness, who allowed me to be a part of it. So I'm going to briefly talk to you about a research study I did based off this conservation study. For a semester project I was required to do in my last year of undergrad. So I'm going to really try to keep it brief and summarize it. It's not too boring, sciencey stuff. But I focused on caudal damage, and that's simply just the injury of, like, the end of the tail is broken on water snakes. So while doing this conservation research, catching hundreds of snakes and measuring them, it was really frequent that some of our measurements we had to put 0 because they didn't have tails. So I thought this was really important since the Nerodia clarkii compressicata is named that for the compressed tail it has to help it swim through the water. So I figured this would definitely affect the way that it lives and the way that it hunts. So that was the focus of my study. This was like a very mini version of what a master's dissertation would be or a PhD type of study, but I had to cram it into 12 weeks and it was for a grade to graduate, so I definitely got a little taste of what it's like to pursue a master's, and it was something I got some pretty interesting results. So I'm just gonna briefly summarize that and go through it as well. Here you can see these photos. On your left is the normal snake with no damages to the tail, and on the other side you can see a specimen that had the tail breakage. So this was really common in the way we were surveying and measuring for these snakes. I thought it was really interesting that almost none of them had tails, a lot of them anyway. So that's what I focused this research on. What I hypothesized was that snakes with the caudal damage would perform less efficiently in the predation. So here you can see a photo in its natural habitat hunting. This is really typical. This is how we would grab them. They're waiting for the fish to come by so they can just strike and get it right there. So that just shows that. And I tried to replicate this in a lab setting, although it was an experiment, so everything was controlled. So some of the methods and the way I set up this study was 16 wild-caught specimens that were from that conservation research, 8 of them which had full tails and 8 that had the caudal damages. So I was testing these 2 groups against each other on how long it took them to consume one fish and then comparing them statistically to see if it was relevant. So I would perform the morphometrics that I previously mentioned on these wild specimens, and then there were fasting periods of 1 week just to make sure that they were all at the same level of hunger per se. I then did videography and direct observation to determine the results of this research, and then I concluded with a t-test statistical analysis. This is a really simple test that's used in a lot of ecology, just whenever you're testing 2 groups against each other, if your results were by chance or if your results were actually relevant. So the number that you get from your data will tell you if it's statistically relevant or not. So I know I talked about palpative induction a little bit before, but this was important for my personal study for this project, just because I wanted them all to be at the same level of hunger. So again, you just stimulate the snake kind of like a toothpaste tube and see if it had been eating anything. So I had to do this for my snakes, and I did keep them in a lab setting, so they were all kept in storage for this amount of time during the study. So I had to perform this palpative induction, and they were all at the same level of hunger. So that was just an important key that I made for this topic. Here you can see there was a huge variety of damage in some of the specimens that I used for this. They weren't all at the same level of damage in their tails. You can see from just a couple centimeters to inches that were off. So this was something not really in my control, and I was under time constraints, so I had to use what I could. But they did have a great variety of these damages. I did their weights, I measured them, and I documented all of these during for the analysis as well. So here's what I used for my replicated lab setting. This was a 180-gallon horse trough that I set up with rock and sand substrate to kind of mimic that edge slope of what they like to hunt in. So I had 0 to 6 inches of water in this area, where 0 would represent the land, 6 represents kind of the most amount of water they're comfortable in. These guys don't like to swim in the deep water. Anytime we would release them and kind of just say, oh, let's put them in the deeper water, they go right back to the edge. So that's where they're comfortable, and that's why I set it up that way. Again, the high vegetation density is definitely their preference, so I tried to replicate that as well. I kept the water heated, as well as stocking it with goldfish and minnows, because again, these guys are documented piscivores. So this is just a photo showing that, and then I used videography, and then I was timing each snake individually on how long it took them to eat one fish. So here's just a photo showing you what I came up with for this project. There were a lot of technicalities that I had to deal with during this time, and this is what I ended up using. I had to change it around a lot, but this is what ended up working. So for the procedure, again, I was just testing these animals individually, but as groups. So the groups with the full tails and the group with the caudal damages. Overall, this was a procedure that looked at the behavior of the snakes, as well as their efficiency and how long it took them to capture one fish. So I was looking for the key interactions between the 3 areas of water and really the snakes. So the 0 inches representing the land, the shallower areas, and then the deepest ends of about 6 inches. I was looking for any behaviors that separated these water depths. Along with these interactions, I definitely took the fish into consideration. They definitely seemed to school and get trapped in the vegetation like they do in the wild, so that's something that worked successfully. And again, it's just something that I was really looking at for key interactions between all of these factors. So I got some really interesting results observing them. So the snakes that had the full tails, they actually were using their tails prehensilely, which just means they use it like another limb. So they're able to grab onto things and kind of anchor themselves. So while I placed each snake in there, the snakes that had the normal tails, they definitely used it, and that's something that I observed through videography and just directly watching them. They are ambush foragers, which I did see in the replicated setting, so that made me feel like, it was naturalistic as it could be, although it was in a lab type of setting. The fish, again, they did get trapped in the density. The ones that didn't seem to, shy away into the deeper water, and then they did seem to school together at times as well. But between the 2 groups, all of the snakes seemed to hunt pretty comfortably within this depth of water from 0 to 6 inches. There were a few specimens that did this very interesting behavior of killing the fish in the water and actually bringing it back to the land to eat it. I observed that in both of the groups, but it was definitely more frequent in the group that had the caudal damages. So that was definitely something that I found interesting, and the tails were definitely used in the predatory behaviors by their prehensile usage. So I wasn't lying, here's some photos. You can see of the 2 groups doing this behavior. So you can see their tails, tried to let it circle right there. Okay. So right here you can see these snakes anchoring themselves. and this one's actually eating. So you can see that they're using their tails while they're hunting. This could potentially be for the current or any other factors for other predators that could be potentially out to get them. It might just also make it easier for them to forage and ambush hunt, I'm not sure. But this was definitely something that I observed in this lab setting that I didn't really get the chance to see while we were doing the conservation study and surveys, because we just tried to grab them as quick as possible. In the second group, here are some examples of them trying to maybe do that behavior, but they weren't quite able to because their tails were damaged. None of them were able to wrap it around and use it the way that the first group did, but 3 out of the 8 specimens brought it back to land to actually eat it. And I think I have a couple of photos of the others. One right here where he brought it back to the land side to eat it. But overall, there were 3 out of the 8 snakes that actually did that behavior. So I thought that was really interesting as well. So here's just some event diagrams summarizing those similarities and differences. The prehensile tail usage, they seem to have less misses. You know, snakes don't have the greatest eyesight, so it takes them a couple times sometimes, but the snakes that had the tail damages definitely missed a lot more from what I observed. Since I did observe each of these snakes, I never just left them and let it record or anything. I was watching all of them. Again, 1 out of the 8 specimens with the full tails brought their fish back to land to actually eat it. When it comes to the caudal damages, 3 out of the 8 specimens did that, so I definitely think it's a relevant behavior. There was a lot more misses, and there was actually one of the specimens I had that had broken bones in its tail. Its tail was kinked almost, so I don't know if that was an injury, but it definitely had a lot of trouble swimming. So these results really told me that their tails are important for predation. Here's some of my data analysis, some simple bar charts that I created. The first chart over here on your left shows the amount of minutes in red right there against the amount of centimeters in tail length that these snakes had. So you can see there's a slight trend line there, whereas the other bar chart is simply comparing the 2 groups of snakes, caudal damage and normal, between the minutes and how long it took to consume one fish. So there wasn't really a trend line there. My one-tailed t-test, which is again just that statistical test that tells you were your results by chance or did they actually matter. 0.5 is the statistically irrelevant number, so if you get any number below that, then that's what tells you, you know, this was significant. I did only run this trial 1 time, so I originally wanted to do it 3 times over the course of the 12 weeks, but I had some technical issues with my setups and some of the snakes that I was keeping, you know, 16 snakes. So although my statistical analysis did tell me it was irrelevant, the behavioral study observations told me that these snakes are definitely using their tails. So along these results, I hypothesized a couple of causes for this tail damage. Since they are living in such a diverse habitat, there are aquatic predators like crabs, there's birds of prey. Usually these damages are accredited to birds, but again, this species is nocturnal, so there's not a lot of birds out there that are hunting the snakes, you know, that I saw during the conservation research where we were surveying, but besides birds of prey, and I've never seen a bird of prey miss. So that was something that was interesting to me, and here are just some photos showing some of the scarring and the damages on some of the specimens I had. Here's the one that had the broken tail. Its bone was crooked, and then these are just scarring, so I thought maybe it could be crabs or some other type of aquatic predator as well, or if they're going into deeper water, it could even maybe be a fish. Another really important topic that while I was researching, I had to do a research paper along with this that had a minimum of 50 sources. So coming across some other scientific papers that are published in scholarly journals. Pseudo Autonomy is just a fancy word for the purposeful tail loss of a reptile. So this is what lizards do. Sometimes, you know, they can regenerate. Snakes, they can't regenerate it, but there are very few snake species that are documented able to purposefully lose their tail. I thought that because the statistical analysis showed that it wasn't very relevant, this could make total sense if they're losing their tail on purpose. When it comes to snakes, the purposeful loss, it requires a lot of force. Their bones actually have to break off, and this is accompanied by a series of physiological reactions, which makes it this behavior of pseudo autonomy, purposeful tail loss. So there are a few snake species that are documented, and I definitely think that more research could be done to study Nerodia clarki, and potentially they have the ability to lose their tail since they're in such a diverse habitat with such diverse predators as well. So this is definitely, it was a mini version of what a dissertation project would be, so further research would need to be done. I wish I could have done further trials to make my statistical analysis a little bit more accurate, but the damage causes could be a further research study because this habitat is so diverse. Again, pseudo autonomy along with habitat overlap and the optimal foraging theory. The fact that these snakes brought their food back to the land to eat it was really interesting to me because the optimal foraging theory just states that the amount of energy an animal has to use to eat is very relevant to the way that they behave. So the fact that they're bringing it back to the land, even though this was a lab setting that was controlled, I think this could be a supportive study to that theory. And in science, a theory is a big deal. It's basically accepted by all biological departments. So I definitely think this research could support that if it was extended into more of a dissertation project. But thank you so much for having me and hearing about my snakes and the Mangrove Saltmarsh snake. Does anybody have any questions?
Any questions, everybody? I have a question.
Have you ever noted lingual luring?
Sorry, what?
Have you ever noted lingual luring?
I read that paper. It hasn't been documented in the wild, but I thought the paper was really interesting, and we definitely, the team that I was with, thought that they could definitely be doing that in the wild.
They do.
Yeah.
Use tongue.
I've produced thousands.
Yeah.
And what they do is they put their tongue out, folding the tips in, protect them, and lure it like a worm for a fish.
Yeah, I found the paper really interesting and the photos that were along with it, but I have never observed it in the wild, or during my study I didn't see that.
Yeah, it's very interesting.
Really cool. Did you have a question?
Yeah, so you said that most of the snakes had damage to their tails, right?
A lot of them, yeah.
What percentage do you think that was, out of the ones that you collected that you saw?
I mean, I would definitely say over 50% in the overall study. We captured hundreds of snakes. But for mine, it was from a couple areas that I just had to take what I could from that night.
Okay.
So that's how I got those specimens.
But overall, I'd say half of them. Or
I would say
over-
definitely over half of, during the conservation study, they have at least a little bit of tail damage. And that's why I really chose that topic.
Were they collected from canal sides, or were they mow the grass?
No, but we did survey some of those areas where we went around stomping on those grasses to hopefully shoo them out. A lot of golf courses and stuff like that have fresh water ponds because this species can't excrete salt. So that's it's a big thing for them where they have to find fresh water ponds where they rely on the rain and also what they eat to get their hydration.
No other questions?